Hello guys, welcome to my YouTube channel. Now today we are going to learn the basics of amino acids. Now this chapter on amino acids is the basic for all biology, biotechnology and biomedical syllabi. So this chapter will help students compete for all the national level entrance exams related to biology. Now first, let us see what are amino acids actually. Now amino acids are the constituents of proteins, right? So these link together to form the peptide bonds. So let us see how an amino acid look like, what the basic structure of amino acid is. Now you can see in the left hand side of the figure, this is the basic structure of amino acids, in which you have the central carbon. This is called as the alpha carbon. Okay, and this carbon has four substituents. One is the variable group, which is called as the R group here. Another is the amino group. This is called as alpha amino group because it is attached to this alpha carbon. And here on the right hand side you can see you have a carboxylic group attached, and this carbon atom carries a hydrogen atom. So this hydrogen atom is called as a alpha hydrogen atom. Now this is the basic structure of amino acids. Now in this way, two amino acids link together to form peptide bond, like first amino acid. So if this is amino acid 1, okay, this so here another, this is amino acid 2, second amino acid. Now these two amino acids form a bond which is called as a peptide bond. So two amino acids join together to form a peptide bond. Likewise, three amino acids, okay, join together to form two peptide bonds. Likewise, we have a chain of amino acids linked together, and that we called as a polypeptide chain. Now how an amino acid forms, you can see here in this figure, this amino acid, first amino acid, and here we have another amino acid. So this is number 1 amino acid, and this is number 2, second amino acid. So these, they are linking together. Here we have water molecule. One water molecule is released by condensation, so this water molecule goes out, okay, and together we have one peptide bond. So two amino acids link together to form one peptide bond. Similarly, we have three amino acids linking together to form three peptide bonds. Likewise, four amino acids link together to form four peptide bonds. For n amino acids, now this is very important for competitive level exams, this formula. So for n amino acids, we have n minus 1 peptide bonds are formed and n minus 1 water molecules are released. So suppose you are asked that how many peptide bonds are formed, will be formed from 8 amino acids. Okay, so here we have 8 amino acids, so they have asked 8 amino acids. So how many peptide bonds will be formed from 8 amino acids? So since we have 8 amino acids, so 7 peptide bonds will be formed and 7 water molecules will be released. Okay, so we will have 7 peptide bonds will be formed and 7 water molecules will be released. So if you are been asked for 9 amino acids, suppose anyone suppose you have been asked how many water molecules will be released from 9 amino acids, then from 9 amino acids will form n minus 1, that is n equal to 9 here. So it will form 8 peptide bonds and 8 water molecules will be released by condensation. So always remember, whenever two amino acids join, one water molecule is released. So if three amino acids join, two water molecules, so n amino acids, we have n minus 1 peptide bonds and n minus 1 water molecules are released. Now let us look at the structure of some common amino acids. So here you see first box describes the structure of some non-polar aliphatic amino acids. So these are non-polar. Why non-polar? So here the R groups, the variable R groups that I talked about, these R groups do not carry any charge. So they are non-polar and they are aliphatic chains. Like you can see the basic structure up to here, the basic structure of all amino acids are the same, only they differ in the presence of the R group, right? For example, in glycine, this R group is a hydrogen atom. In alanine, this R group is a methyl group. In proline, this R group is a complex cyclic group. This in valine, this R group is a isopropyl group. Like that, we have leucine, this R group. We have isobutyl group. Here we have the structure of isoleucine, and likewise you will have the structure of methionine. Now, one thing to notice here is in glycine, you have simple hydrogen atom as the R group. In alanine, you have a methyl group, but in proline, you have a cyclic group as the R group. So this is a cyclic group. So questions can be there. Questions can be asked that which amino acid has a cyclic group, or which non-polar amino acid has a cyclic group. So answer will be proline. Likewise, methionine, you can see, so the R group carries a sulfur in its chain, so it is a sulfur containing amino acid. Now let's come to the second box where we describe the structure of some aromatic amino acids. You will find here there are three aromatic amino acids: phenylalanine, tyrosine, and tryptophan. In phenylalanine, we have the methylene group, CH2 group attached to a benzene ring. In tyrosine, we have a methylene group as well as an OH group attached to the benzene ring. And tryptophan, this is a complex structure as the R group. Now we come to the third box where we describe the structure of polar uncharged R groups. Here we have, so these are polar uncharged, that means the R groups are polar, but they do not carry any negative charge or positive charge or whatever charge, but they are polar due to the presence of polar groups. Like in serine, we have a CH2 group attached to an OH group. Due to this presence of this OH group, this serine amino acid becomes polar. Likewise, in threonine also, you have an OH group. So because of the presence of OH group, threonine amino acid becomes polar. In cysteine, you have a CH2 group attached to a thiosulfate group, that is SH group. So questions can be asked from here: which amino acid, which polar amino acid, uncharged amino acid carries a thiosulfate group as the R group? So answer will be cysteine. So next we have asparagine. This amino acid is also polar due to the presence of this NH2 group. And then we have a glutamine. This amino acid is also polar due to the presence of this NH2 group, but they do not, all of the 5 amino do not carry any charge, whether positive or negative. So that's why they are polar but uncharged. So they are polar but uncharged amino acids. Now we come to the positively charged R groups. So here you can see the R groups, okay, they carry a charge. So that's why we have they carry positive charge. So these three amino acids, lysine, arginine, and histidine, so these three amino acids are polar and positively charged R groups, right? So here you can see in the backbone of the R group, we have this NH3 plus group carries a positive charge. This NH2 group, okay, this NH2 group, this NH2 plus group carries a positive charge, and here also this NH3 group carries a positive charge. Now we come to another amino acid set of amino acids with a negatively charged. So these are called as aspartate and glutamate. So these carry a negative charge at the CO minus group, and these carry a negative charge at the CO minus group, right? Now we have talked about the structure of the non-polar aliphatic R groups, as I have talked about these 5 amino acids. So in this category we have actually 7 amino acids, okay, so these which are non-polar and aliphatic, R groups. So these 7 amino acids are non-polar amino acids, because you can see this NH3 plus group has a charge of +1, okay, and this CO minus group has a charge of -1. So together the whole charge +1 -1 cancels, and together net charge becomes 0, because you don't have any charge on this R group. So in this category of non-polar aliphatic R groups, you have all total 7 amino acids which do not carry any charge. In this category of aromatic amino acids, you have all total 3 aromatic amino acids, but these aromatic amino acids also do not carry any charge. Next category, if we come to here, polar uncharged R groups. So here we have all total 5 amino acids, but again these amino acids do not carry any charge, because you can see NH3 plus, the positive charge of NH3 plus group is balanced by the negative charge of CO minus group, and the R group do not carry any, R group does not carry any charge on its main chain. So that's why all total overall charge, the overall charge becomes as a result the overall amino acid becomes chargeless, so you do not have any charge. Here, in case of positively charged R groups, this category you have all total 3 amino acids which have positively charged. You can see here, if we talk over here, the plus 1 charge of NH3 plus group is balanced by the minus 1 charge of CO minus group. So all total here the net charge becomes 0. But here we have, in case of lysine, this NH3 plus group carries a positive charge of plus 1. So overall charge of lysine becomes plus 1. Similarly, in case of arginine, NH3 plus the group, the positive charge of NH3 plus group is balanced by the negative charge of CO minus group. But here we have a positive charge of plus 1 on arginine. Okay, so overall charge of arginine becomes positively charged. So in this category we have 3 amino acids which are positively charged. Again we have another category of negatively charged amino acids. So here in case of aspartate, you can see positive charge of NH3 plus group is balanced by the negative charge of CO minus group. But in case of the CO minus group, we have another CO minus group in the as the R group. Here we have a net charge of minus 1. So overall charge of aspartate becomes minus 1. Likewise, in glutamate also, overall charge of glutamate becomes minus 1. So these positively charged R groups, when you place in an electric field, okay, so this migrate to the negatively charged electrode or the cathode. Okay, so questions are sometimes asked that, say, with these amino acids will migrate to which electrode, right? So questions are sometimes asked here, and this negatively charged amino acids, when placed in an electric field, these migrate to the positively charged electrode or the anode. Now this cysteine amino acids which we have talked about, this carry a thiosulfate group in its R as in its main chain as the in the R group. So these two thiosulfate groups condense with the release of water in presence of oxygen. Okay, so 2 hydrogen atoms and 1 half O2, that means 1 oxygen atom, these together form water and they leave, giving together this disulfide bonded cysteine residues. So here, so that's how one disulfide bonded amino acid form. Now there are certain one letter abbreviation of amino acids, right? So let's look at the one letter abbreviation of amino acids. In case of glycine, if you see, the abbreviation is G. Now this topic, this one letter abbreviation of amino acids is very important for the all the GATE, JAM and the CSIR NET exams. So this the one letter abbreviation of amino acid. Now let us come to the one letter abbreviation of amino acids. So this topic is very important for all the competitive national level exams like GATE, JAM, CSIR NET, DBT. Questions are asked sometimes on the one letter abbreviation of amino acid. So if we talk about glycine, so it has the one letter abbreviation of G, alanine has A, proline has P, valine has V, and leucine has L. Isoleucine has I and methionine has M. So these 7 amino acids are all non-polar aliphatic R groups. So in case of phenylalanine, it is the abbreviation is F. In case of tyrosine, the abbreviation is Y, and in case of tryptophan, the abbreviation is W. So these three abbreviations are very important. Okay, in case of serine, the abbreviation is T. In case of threonine, the abbreviation is T. In case of cysteine, the abbreviation is C. In case of asparagine, abbreviation is N. In case of glutamine, the abbreviation is Q. Again, these two abbreviations are very important. So in case of lysine, we have the abbreviation is K. In case of histidine, the abbreviation is H. In case of arginine, abbreviation is R. In case of aspartate, abbreviation is D. And in case of glutamate, the abbreviation is E. So again, these three abbreviations are very important. Now we come to the UV light absorption by some aromatic amino acids. But first, let us talk about the Lamberts BS law. So Lambert BS law, we have the formula of A is equal to log I naught by I is equal to epsilon naught Cl, where A is the absorbance, I naught is the intensity of the incident light, I is the intensity of the transmitted beam, epsilon is the molar extinction coefficient, C is the concentration of the sample, and L is the path length. So though we will have a separate video on Lamberts BS law, but to just have a quick recapitulation on it. So if you have a light source, say a lamp, and if you pass it through a monochromator to select a particular wavelength, okay, and then if you, this is the wavelength that you are, that is falling, okay, this is wavelength that is falling on the sample. So it has an intensity, it has an intensity of I naught, okay, it has an intensity of I naught. Now if you allow to fall this intensity or the wavelength of the light on the sample, okay, certain part of the light is absorbed and certain part of the light comes out from the sample. Okay, so this, the intensity that comes out from the sample is designated as I. So the initial intensity that is falling on the sample is I naught, and the intensity of light which is coming out on the sample after absorption is the I. The sample has a concentration of C moles per liter, and you detect that with a recorder, the how much of the intensity of light is absorbed and how much of it is not absorbed. So then you have placed the values in the formula, log I naught by I is equal to epsilon Cl, and then you calculate the absorbance, that is how much the absorbance has been carried out. There are three amino acids, aromatic amino acids, that we have talked about: phenylalanine, tyrosine, tryptophan. It has been seen that these three amino acids, okay, absorb the light samples at a wavelength of 280 nanometer. Okay, so these three amino acids are the only amino acids which absorb light at 280 nanometer wavelength. So when you allow to fall light sample of 280 nanometer wavelength on a sample containing any of the three amino acids or all of them, then it has been seen that they absorb light only at this particular wavelength, okay, called as the 280 nanometer. If you can see in the graph, tryptophan absorbs the highest at the 280 nanometer. So this is the it has a peak at around somewhat around 280 nanometer. Okay, so then we have the tyrosine, and then the phenylalanine absorbs the lowest at 280 nanometer. So tryptophan absorbs the highest at 280 nanometer. Then we have tyrosine, and then we have the phenylalanine at 280 nanometer. Now this topic is very important for the competitive exams like GATE, JAM, CSIR NET, BET, etc. Those who are eyeing for these competitive exams. There are some uncommon amino acids. So the first uncommon amino acid which we need to know is called as the 4-hydroxyproline. Now this amino acid is actually a derivative of proline, and it is found in the plant cell walls and collagen. So there are two sources. First, we have plant cell wall, so it is a constituent of plant cell wall, and second, it is constituent of the collagen. Then we have another amino acid called as the 5-hydroxylysine, which is a derivative of lysine, and it is also found in collagen. So this is the source. Okay, so next we have N6N-methyl lysine. As you can see, get from the name, it is a derivative of lysine, and it is found in the muscle protein myosin. So this is the source. Okay, then we have another amino acid called as a gamma carboxyglutamate. So this actually a derivative of glutamate, as you can figure out from the name, and it is found in the blood clotting protein prothrombin. So there are two sources. First is the blood clotting protein prothrombin, and in certain other proteins that bind calcium ions as a part of their biological function. So there are certain other proteins which need calcium ions for the proper function. So gamma carboxyglutamate, this amino acid, actually a derivatized amino acid, this amino acid is also found in those proteins as well. Next we have selenocysteine. So this is derived from serine and contains selenium rather than the sulfur of cysteine. So this is actually what happens, in selenocysteine. This is actually a derivative of serine, and instead of the sulfur of cysteine that we have just looked before, the cysteine contains sulfur, you have selenium. Now this is very important because many questions have been asked in various competitive exams on selenocysteine. You have to remember that it is derived from serine and it contains selenium. It does not contain the sulfur, but in place of the sulfur of cysteine, it contains the selenium. Another amino acid we have pyrrolysine. It is found in certain methanogenic archaebacteria. There are two more amino acids. Here uncommon amino acids. Number one is the ornithine and number two is the citrulline. These two amino acids are mostly found in the urea cycle and in the biosynthesis of arginine. Now these amino acids, okay, if we talk about them, these amino acids can also act as acids and bases. How? Let's look at them. So first, if we see the structure of this amino acid, you have the positive charge of this NH3 plus group has a positive charge of +1. This COH group has a positive charge of 0. So altogether the overall charge becomes +1. Okay. At the neutral page at pH 7, okay, this NH3 group loses the positive charge. Okay, sorry, this carboxylic group loses the one proton and it becomes negative charge. So the charge on carboxylic group becomes -1 and the charge on the NH3 plus group remains as +1. So overall the net charge becomes 0. And again, now on the second time, this NH3 plus group loses the proton, and here the charge on NH2 group is 0 and the charge on CO minus group is -1. So the overall charge becomes -1. So first what happens, this carboxylic group loses protons. So in the first case, this carboxylic group loses protons. So proton comes out from this carboxylic group in the first case. Afterwards, if you continue to increase the pH, for the second time this amino group loses the proton. So first the carboxylic group loses the proton of the first time during the initial low pH. We have the carboxylic group loses the proton, and we continue to add the as a base. Okay, so this NH3 group next loses the proton. Okay, so here so we know that if you add H plus or if you accept H plus, a compound that accepts H plus, okay, that compound behaves as a base. Okay, so and if you release H plus, okay, so if you release H plus, then that compound behaves as an acid. So here you can see in this Voet ionic form, if this zwitterionic form reverses back, then how it will reverse? It will take a proton, okay, it will accept a proton and it will revert back to this structure, the first structure. So by accepting a proton, here it is behaving like a base, okay, or it may release this proton, okay, and then it can revert to this structure of -1, and then by that time it will act as an acid. So it depends upon the medium condition whether the amino acid will act as a base or the amino acid will act as an acid. Now I have already talked about that there are two ionizable groups of amino acid, NH3 plus group and the COH group. Now whether the R group will have an ionizable group or a non-ionizable group depends upon the R group. We are mostly talking about the general structure till now. Later on we will be dealing with structures of amino acids in which R group is also there, but excluding the R group we have two ionizable groups, mostly NH3 plus group and the COH group. Now this is the non-ionic form. Here you will see this NH2 group has a negative as charge of 0, this COH group has a charge of 0, so overall charge is 0. But here we have the zwitterionic form where we have equal charge, okay, this has a charge of +1, this has a charge of -1, so overall charge is 0, and this exists at a pH value of 7. Okay, so now this zwitterionic form can act as an acid by releasing the proton, or it can act as a base by accepting the proton, which I have already talked before. So it depends upon the medium condition whether it will release proton or it will accept the proton. Okay, here it is releasing proton, so it is acting as an acid, and here it is accepting a proton, so it is acting as a base. So it depends upon the medium condition totally whether the amino acid will accept the proton or it will release the proton. Accordingly the amino acid will act as an acid if it accepts the proton, if it releases the proton, and it will act as a base if it accepts the proton. Right, now let us look at the titration curve of glycine. So what happens here is that you have an amino acid, and if you continue to add a base and you continuously add the base, you increase the pH, and if you draw the titration curve you will have this characteristic curve as shown here. So what happens, at low pH values, if you look at the structure of glycine, here we have no charge on the carboxylic group and +1 charge on the amino acid group, NH3 plus group. So at low pH values it is positively charged. Okay, so at low pH values it is positively charged. Okay, so at the zwitterionic form it is charge is zero charge, because the plus charge of NH3 plus group is balanced by the equal and opposite charge of CO minus group, and at high pH values you have a minus charge of 1, because NH2 group doesn't carry any charge, and the CO group carries a negative charge of -1. So at high pH values it is negatively charged, right? So this is actually attributable to positively charged form of glycine is attributable to low pH value, and the negatively charged form is attributable to high pH value, and the zwitterionic form is attributable to the neutral pH value, right? So here you can see up to here, this portion from here to here, okay, this we have a low pH value. So here predominantly what kind of the form of glycine prevails? Here we have negatively charged glycine. Okay, this is the neutral pH value. Here we have this is the neutral pH value where the zwitterionic form is prevalent, and this is actually the high pH value, and here we have the positively charged sorry, this will be the positively charged and this will be the negatively charged. Here we have the negatively charged form of glycine prevails. Okay, here we have the negatively charged form of glycine prevails. Now here we, as you can see, there are two regions of buffering power. Buffering power means here in the buffering region you have more or less constant pH. So from here to here, if you observe the titration curve of glycine, from here to here one buffering region is present. More or less there is very much small increase in pH in comparison to the very large increase in the base. Okay, you from here to here you will see here we have a, you have added a very much amount of base or OH minus ions equivalence, right? But here in comparison to that, you have at each stage there is only a very small increase in pH. So this is the first region of buffering power, and from again here to here there is another region of buffering power. So glycine has two regions of buffering power. In these two regions there is very small increase in pH in comparison to the huge amount of addition of base. So one thing in which questions are asked, that if you have an amino acid, okay, the pi value is given, so and if the given pH, okay, is less than pi, then the positively charged form prevails. Okay, positively charged form prevails of the amino acid. But if the pH is greater than pi, okay, then the negatively charged form prevails. Okay, then a negatively charged form prevails. Suppose pi value is given to be, say, of an amino acid is given to be 6. Okay, pi value is given to be 6 and the pH value is given to be, say, 2. pH value is given to be, say, 2. Here, since pH is less than pi, so obviously the positively charged the form of the amino acid will prevail, and what will happen, you have a positively charged amino acid here. You have positively charged amino acid here, so it will migrate to the cathode. Okay, it will migrate to the cathode. Suppose pH is suppose pi is given to be 6. Okay, pi is given to be, say, 6 and pH is given to be, say, 10. So this amino, since pH is greater than pi, so this amino acid will be negatively charged and it will migrate to the anode. So same amino acid is behaving in different way, and because of the medium condition. So whenever you are lowering the pH below the pi, that amino acid is becoming positively charged and it will migrate to the cathode. But that same amino acid, if you increase the pH beyond the pi of the amino acid, that amino acid will behave as a negatively charged amino acid and it will migrate to the anode. Why? Because at low pH values, I have already talked about this, the amino group is protonated but the carboxylic group doesn't carry any charge, so overall charge becomes positive of +1. So that's why at low pH value the amino acid is acting as a positively charged amino acid and it will migrate to the negatively charged electrode, that is cathode. And as you continuously increase the pH beyond the pi of the amino acid, what happens, the amino group doesn't carry any charge over it, but the CO minus group has a net negative charge of -1, so overall charge of amino acid becomes -1. So the amino acid then becomes a negatively charged amino acid and it will migrate towards the positive electrode, that is anode. Okay, So we have already said that the two ionizable groups are glycine, because the R group in case of glycine is a hydrogen atom, so so it doesn't carry any charge. So two ionizable groups of glycine are titrated with a strong base such as NaOH. Whenever you titrate an acid or a compound with a base, you have a titration curve, characteristic titration curve, and more specifically a weak acid one. So if you titrate a weak acid with a base, then you have a titration curve as shown in the left hand side of the figure. Okay, compare it with the titration curve of acetic acid that you get while titrating it with NaOH. You will get very much similarity in the titration curve of acetic acid and glycine. So this plot has two distinct stages, and these correspond to the deprotonation of two different groups. So in the first stage I have already talked about, this first stage corresponds to the deprotonation of the carboxylic group, and this second stage corresponds to the deprotonation of the amino group. Okay, so now at very low pH values, at low pH value what happens? Okay, this NH3 plus group that has a charge on it, but COH group doesn't carry any charge, so we have a positively charged amino acid. Okay, then the carboxylic group, as you increase the pH, the carboxylic group first loses its proton, and the amino acid at the pH is equal to pi. Okay, that is this becomes the at pH is equal to 7, this amino acid becomes the zwitterionic form. It doesn't carry the positively charged R group, or positive charge group on NH3 plus group balances the negative charge on CO minus group. Okay, so then it becomes a zwitterionic form, and if you continue to increase the pH, what happens? Then the amino group loses its proton. So okay, and then the amino group loses its proton and the amino acid becomes negatively charged. So first happens from 0. If you can see from 0, as you continuously increase the addition of base NaOH, first the carboxylic group loses its proton, and it continues to rise up to pH is equal to 7, when you have equal contribution of the positively charged R group and the negatively charged COH group. And then if you continue to add the base, it increases, then the amino group loses its proton and you have the negative charge amino acid, and the titration is essentially complete as around pH, say pH is equal to 12. Okay, now glycine was a neutral amino acid. Now let us look at the titration curve of, say, another negatively charged amino acid. So glutamate is a negatively charged amino acid, right? Glutamate is a negatively charged amino acid. As you can see, at low pH values, if you see, this glutamate has a positive charge. Okay, at low pH values, this glutamate has a positive charge at the pH is equal to 7. pH 7, it has a zwitterionic form where you have no charge, and at high pH values, first the glutamate becomes the charge of carries a negative charge of -1, and then as you continuously increase, it has a negative charge of -2, right? So here we have at low pH values, you can see these two carboxylic group do not carry any charge on them, only the amino group carry a positive charge of +1, so overall charge becomes +1. As you continuously increase, one of the carboxylic group loses a proton. Okay, and then you have CO minus group carries a charge of -1, this amino group carries a charge of +1, and here we have no charge, 0 charge on this carboxylic group, so overall charge becomes 0. As you continuously increase, then this carboxylic group loses a proton, this CO minus group carries a charge of -1, this CO minus group already had a charge of -1, this the NH3 plus group has a charge of +1, so overall charge becomes -2 + 1, so -1. And then as you increase this, the NH3 plus group loses a proton. Okay, now now you sequence this CO minus group had a charge of -1, this CO minus group had a charge of -1, and this NH2 group doesn't carry any charge, so overall charge becomes -2. So as you can see, as you continuously increase the addition of base, first the one carboxylic group loses the proton, then another carboxylic group loses the proton, and finally at high pH values this increases. Ultimately at high pH value the amino group loses the proton and the titration is complete. So here you can see, here we have one pK1 at this point. Okay, one pK1 at this point, one pKr. This pKr comes here because there in the previous case there was just hydrogen as R group, but here in case of in case of glutamate you have the CH2 COH. So this is the R group here. So because of the presence of R group you will get another, this is R group here. So because of the presence of R group you will get another point in the titration curve, and then we have the pK2. Before we move now, if you look at, observe the titration curve of glycine, pi, how will you find the pi? Pi is the average of pK1 plus pK2 by 2. Okay, so pK1 plus pK2 by 2. So this is the average of the pK1 pK2 by 2. So this is the pi of glycine, because in glycine the R group is the simplest, which is hydrogen. But in case of glutamate, if you observe, the R group is not as that simple as that of glycine. You have a complex R group of CH2COH. So you will get another point here, which is called as a pKr. Now as you can see, pKr is intermediate between pK1 and pK2. So pKr lies more or less at the midpoint of pK1 plus pK2. Okay. Now and this pi, so how will you find the pKr? This pKr is given by, it is the average of pK1 and pK2. And then if you are asked to find the pi, because in case of negatively charged amino acid the pi lies between pK1 and pKr, so pi will be the average of pK1 and pKr, right? Okay, so because pi lies between pK1 and pKr. Now if you observe the titration curve of histidine, the R group, this is the R group here, and this R group is a complex group. So this is the R group that you have. In case of at low pH values, this NH3 plus group carries a positive charge of +1. This carboxylic group doesn't carry a charge on it. Okay, so this has a 0 charge, and this also has a positive charge of +1. Here we have a positive charge of +1. So the overall charge is +2 at low pH values. Okay, now as you increase the pH, first this carboxylic group loses a proton, and it loses a proton and overall charge becomes +1. Then you increase the pH, this is a sweet ionic form, overall charge becomes 0, and then as you continuously increase the pH, the overall charge becomes -1. So as you can see here in the graph, it increases after continuous addition of base. First the carboxylic group loses a proton, okay, then the in pKr, when it reaches the where pH is equal to pKr, the R group loses the proton, and then the amino group at high pH values, amino group loses the proton, right? Here in now this is, as you know, this is a positively charged amino acid. So here as you can see, pKr, in case of positively charged amino acid, pKr like previously lies between pK1 and pK2. So pKr lies between pK1 and pK2. This is the pK1 and it lies between pK2. Okay, so pKr is given as pKr is given as pK1 plus pK2 by 2. Okay, so but here pi lies between, unlike the previous one in glutamate where the pi used to lie between pK1 and pKr, here in case of positively charged amino acid, this pi lies between pKr and the pK2. Okay, this pi lies between pKr and pK2. So pi value is given as pKr plus pK2 by 2. Okay, now there are some chemical environment effect on the value of pK. Likewise, if you see in case of acetic acid, okay, in case of acetic acid, this CH3 group, okay, in case of acetic acid, if we first talk about a simple acid, say acetic acid, weak acid, say acetic acid, CH3 group has a plus I effect. So it has a tendency to lose the proton and become stable, okay, and become stable. In case of methylamine, this CH3 group has a plus I effect, so it has a tendency of, this NH3 plus group has a tendency of losing a proton to become stable. But in case of alpha amino acid, it has been observed that the pK values are somewhat greater than in the pK values of any acidic acid, any acid or a base. Why? Because this CH2 group, CH2 group has a plus I effect, but this NH3 group also has a minus I effect. Okay, this NH3 group has a strong minus I effect. So because of the strong minus I effect, this tendency of the amino acid to lose a proton and behave as an acid becomes 1000 times more than in case of any other acid, as compared to any other acid. Okay, so because of the presence of this amino group, which has which has a minus I effect, okay, on in any amino acid, so the tendency of any amino acid to behave as an acid becomes 1000 times more than as compared to any other weak acid, say acetic acid. Now let us talk about some biologically important derivatized amino acid. So first we have gamma aminobutyric acid, which is often abbreviated as GABA. and we have another amino acid which derivatized amino acid which is dopamine, which is a tyrosine derivative. So GABA is actually a glutamine derivative. This is very important. So GABA is actually a glutamine derivative. Okay, so, and dopamine is actually a tyrosine derivative. So these two amino acids are important as neurotransmitters. Neurotransmitters, I think you know, which are substances which are released by nerve cells to alter the behavior of the neighboring cells. So these two are neurotransmitters. These are actually derivatives of some amino acids. Another amino acid we have histamine. This histamine is actually a derivative of histidine, and it is a potent local mediator of allergic reactions. Another derivative is amino acid we can see is, you can see, thyroxine, which is actually a derivative of tyrosine, and it is actually an iodine containing thyroid hormone. It is a very important thyroid hormone. So another tripeptide we have, which is a glutathione. This is actually a tripeptide. As you can see, it is actually a tripeptide of 3 amino acid. Okay, glutamine, cysteine, and glycine. Okay, these 3 amino acids join together to form a glutathione. Okay, these 3 amino acids join together to form the glutathione. Now this glutathione, this tripeptide, is also important as a derivative of amino acids. Here you can see this is the structure of gamma amino butyric acid. You can see the gamma carbon. Okay, the carboxyl group is joined to the carbon. That carbon we call this alpha carbon. Next to the alpha carbon we have the beta carbon, and next to the beta carbon we have the gamma carbon. So this is the gamma carbon. Okay, and this gamma carbon carries the NH3 plus group. Here we have the structure of histamine. As you can see, it has a very similarity to the structure of histidine. Here we have the structure of dopamine, and here it is the structure of thyroxine. So here in case of thyroxine, this I can say, I hope you can figure out, it has this group is very similar to the group R group present in tyrosine. That is why this thyroxine is derivative of tyrosine. Now on the right hand side you can see this glutathione. Okay, so how does a glutathione link? Two glutathione molecules link together. Okay, two glutathione molecules link together by the release of one water molecule to form a chain of amino acid. Okay, so this is the chain, and these two glutathione molecules link together to form this glutathione disulfide GSSG. Okay, by the release of one water molecule. So last we will talk about one of the important application of this amino acid that we can see here. We will talk about the green fluorescent protein. Now GFP, green fluorescent protein, is actually used as a reporter enzyme or reporter gene. So what happens? Suppose you have engineered a protein. You have engineered a protein, and you link this protein to, and you attach this engineered protein to a green fluorescent protein gene. Okay, so this is actually GFP. Suppose you engineered a protein, okay, and this link the gene of the engineered protein with a GFP gene. So this is GFP gene, and you see the expression level. So if the expression level, if the protein is expressed, if this protein is expressed, you will observe you will observe green fluorescence. So that is an indication that your protein, your desired protein, has been expressed. If this is expressed, you will observe green fluorescence. If this protein is expressed, or if this gene is expressed, you will observe the green fluorescence. Why? Because this protein is attached with this GFP gene. Okay, if this protein is attached with the GFP gene, so if this protein is expressed, then automatically this GFP gene will also be expressed, and this GFP protein will produce what? This GFP protein will produce GFP gene. This GFP gene will produce what? GFP protein, and this this recombinant protein gene will also be expressed, and you will have a product of, if after expression you will have the product of, you will have your desired protein. This is your desired protein. Okay, this is your desired protein, and here you will have the GFP protein. This is the GFP protein. So once it is expressed, so if your desired recombinant protein gene is expressed, then you will have, because these two are attached, you have attached the recombinant protein with the GFP gene, recombinant protein gene with the GFP gene. So these two will be expressed simultaneously. So if the recombinant protein gene is expressed, GFP gene will also be expressed, and then you will have, once they are expressed, we will have the recombinant protein along with the GFP protein. This GFP protein will give green fluorescence under UV light. So this GFP gene is mostly used as a reporter gene by attaching it with a protein coding gene, and these two will have to be expressed simultaneously. So once the protein encoding gene expresses and forms the protein desired protein, GFP gene is also expressed and produces the GFP, green fluorescent protein, and together the green fluorescent protein, after expression, green fluorescent protein will produce the green fluorescence, and by the amount of fluorescence you can figure out how much the protein, your desired protein, has been expressed. You can figure out the level of expression. So it is found naturally in the bioluminescent jellyfish Aquaria victoria, and this is very important, it has been asked in many competitive exams. The fluorophore contains three amino acids: serine, tyrosine and glycine. So as you can see in the right hand side, this is the serine, this tyrosine, and this is the glycine. So these three amino acids come together, then they undergo cyclization followed by elimination of water to form the final fluorophore. So this is the fluorophore of the green fluorescent protein, and this fluorophore is actually responsible for producing the green light. So here you can see, this is how the jellyfish Aquaria victoria looks like under UV light, because it has the green fluorescent protein within inside. So when you illuminate that jellyfish under UV light, so it glows and gives green color.